One character, three important laws what the Mendel brought out: law of segregation, law of independent assortment, and law of dominance. That is known. And what all the characters he has to been used for study, like the flower color, purple and white, um, peace, right? And then that's what he found out. If he crossed the two plants, one plant with the purple color and another plant with the white flower color, and he got a hybrid F1. That was the purple color. He decided. I mean, he concluded that the purple is dominant over the white. So purple is controlled by one allele, and white is controlled by another allele. Um, uh, for this gene, two alleles are there, and the dominant allele will suppress the expression of the recessive allele. That's why in F1 you are getting only purple color. The flower position. Similarly, another character what we have chosen for his experiment: axial, where the leaves meet stem, the terminal at the end of the palm. So the axial or the terminal, right? Flowers are appeared in the axis and flowers are appeared in the terminal. Now in tomato you say that the determinant and indeterminate. So this kind of again how the genetics is there. You have to study and conclude. But here in bees he got this character and he could able to judge easily. So seed color, yellow color and green color. Yellow is dominant over the green, and the flower position is axial is dominant over the terminal. And then the seed shape, round and wrinkled, round dominant over the wrinkle, pod shape, green dominant over the yellow, pod color, smooth is dominant over the constricted, stem length, that is tall, is dominant over the dwarf. So it's, yes, F1 results of Mendels dihybrid crosses. All plants displayed the dominant form of both. All plants inherited one allele for each trait from each parent. All plants were heterozygous. And yes, you got, it again, if it starts segregating, you can just conclude that. So seed shape, how the parent was there, and then the hybrid, what was observed, whatever is explained in the first table, same thing, he could get it. Easily one can understand. Otherwise, it was totally a new thinking at that time, and nobody could, took him seriously for almost like 30 years, 30, 40 years, 30 years. Even this is all explained by him. Yeah, this is how the alleles, if you take the parents, it is a male parent and female parent. This is, like if you take it as a female parent and this is the male parent. If you take two genes, then there are four alleles. Each gene, two alleles, or dominant and recessive. So capital letter represents the dominant and the small letter recessive. Similarly, for Y, it's dominant and recessive. So you will you will get a grouping. They assort independent. The segregate independently. They're assorting, independently. But yes, possibility. If this is the case, first gene you can have dominant, and the second gene dominant. First gene dominant, second gene recessive. First gene recessive, second gene dominant. First gene recessive, second gene also. So that means four possibilities of formation of the gamete. These are the gametes. Gamete 1, gamete 2, gamete 3. Similarly, whatever you find in female parent, the male also will get the same kind of gametes formation. Now these gametes, that means if you cross the flower, the pollen you are placing on the stigmatic surface, that's a gamete that will enter into the embryo sac where egg cell is there. There's a female gamete, and that female gamete we are what we are referring here. So different types of gametes you can get, right? So for that you may need to have this possibility of grouping and possibility of formation of zygote. This is a zygote, right? So from the female also dominant, both the alleles are dominant, and male also dominant, you will end up with homozygous dominant for both the genes. Like that, all possible combinations of zygote formation, right? So this understanding is very important. Otherwise, you would not have arrived at, the how you can plan for breeding a particular character manipulation, right? So that's why this is very important. So here, uh, the characters they took yellow, round, green, wrinkled, the seeds, color and shape, they have taken it together. Sometimes this same character also can be controlled by two genes. Then you will have four different morphotypes are phenotypically appearing, and I have plenty of examples. So this is what the formation I think you have realized. So that means to become a just a minute. Okay, this is what the independent assortment of genes. If you take two different genes, they are freely. If there is any restriction, linkage is there, then you cannot find all these four types of gametes. Remember, that's what linkage. Whenever you mean those two genes alleles will try to go together, maybe the proximity or whatever reason. But if the independent assortment means they're grouping separately, freely, right? Segregation is just getting into, whereas grouping is, formation of, combinations of genes, you can say. Similarly, it depends on the chromosome also. Okay. So that is, uh, the second and third law, principle of dominance. Some alleles are dominant and others recessive. Mendel wanted to know if the recessive alleles disappeared or they're still in the F1, just hidden. How, where that allele has gone? Already if everything is, the found out now for you may not be, the surprise. For him, it was a surprise where it has gone. It's not appeared. So that's why that was appeared in the F2. So it was hidden in F1, letter recessive, whereas it is appeared in F2 because of that getting into homozygous recessive condition. So this is what, principles of dominancy has confirmed. Right, yeah, there is a recessive allele which is being dominated, which is not being allowed for its expression because of the dominant. Um, Trusted. Dominant relation, complete dominance. Now the dominance also, it can be complete dominance and incomplete dominance. Always heterosis breeding will term into, whether complete dominance are there or incomplete dominance are there. So incomplete dominance, it's the complete dominance means it's a homozygous dominant is same as the heterozygous. Whereas incomplete dominance means the heterozygous phenotype is somewhere between that of the two homozygous. So homozygous dominant and homozygous recessive two phenotypes are there. If it is incomplete, it will not be, what we said it, the only the pink is dominant means F1 will get to. But here, if it is incomplete, shade of that pink. So that is what incomplete. Then codominance. So non-identical alleles specify two phenotypes that are both expressed in heterozygous condition. That means, the homozygous dominant will have one phenotype and homozygous recessive will have another, whereas in between, that is heterozygous, will be a different phenotype. So flower color in snapdragon, incomplete dominance, red flowered plant, white flowered plant. If both are homozygous, only pink flowered you will get the F1. That is, that means heterozygous condition, instead like the red or white, it is in between. The pink you will get. That's heterozygous. So that becomes incomplete dominance. Okay. A red flower, two alleles, allow them to make a red pigment. Why white flowers, two mutant alleles can't make a red pigment, that is white. Pink flowered have one normal and one mutant allele, make a smaller amount of red pigment, so it becomes pink. That's how physiologically or biochemically you can also explain this. Yeah, this is what incomplete dominance in flower color. If the parents are, one parent is white, another parent is red, whereas your hybrid F1 is going to have all the plants, you know, pink only. If you self, then F2 you will get all colors. Flower current, incomplete dominance. I mean, pink flowered plant and pink, okay, the same thing. They had. Its ratio is being explained here. F2 ratio is 1:2:1. So one is uh, white, homozygous recessive. One is red, homozygous dominant. These two are heterozygous, right? Heterozygous, resulting into pink. So that's what incomplete dominance examples you can just it is given here. Neither allele is dominant over the other. Combination of red and white flowers is very well explained. Codominant, one more is sickle cell. That's one more the conditions, how the alleles are interacting. So, so it is called everything what we are explaining is the intra-allelic interaction, right? At one location, whatever gene is there, the interaction between the two alleles present in the genotype. So that is, the first we said it dominant, and then we said it incomplete dominance. Then the third one, we are saying the codominant sickle cell disease, AB and O blood group in humans. It is a codominant, right? Both the alleles have got their expression. Incomplete is the quantum of production, whereas here it's totally a different one, right? Codominant, even our self compatibility also codominant kind is observed in cabbage. So that is what codominant you should understand, right? Then the pleiotropy. Alleles at a single locus may have effects of two or more traits. It's one more confusion is being added. Pleiotropy. Always we said it, one gene controls one character. Whereas here, the alleles at a single locus, that means one gene will have the effects on two or more traits, right? So same gene is controlling two characters like that, more than two characters. Classical example is the effects of the mutant allele at the beta globin locus that give rise to a sickle cell anemia. So this is a pleiotrophic effect example, right? Some kind of intra-allelic interactions. You said it, same locus, alleles, how they are interacting. Now this epistatus, it is now not the same locus. It is two different genes. How there is interaction. The interaction between the products of gene pairs, the two different genes, how they are interacting. Common among the genes for the hair color in mammals, okay? And we get in plants plenty. So comb shape in poultry, walnut comb, then the rose comb and the pea comb. So single comb and the different kinds of, I think when we study the backcross breeding will throw some light on this, what are the different digenic ratios. That is all because of the epistatis only. Interaction between the epistatis is it's non-allelic gene interaction, right? That means two different genes or more than two also. This is just example. Uh, when multiple genes are there, then continuous variation you will get. So greater the number of genes and the environmental factor influence is more. Blurring effect, whether it is because of the genes or it is because of the environment, it's difficult to bring it out. But that is what your population genetics, country genetics deals with. We don't study their one gene, two gene. It is, start calling them additive, dominance, and epistatic kind of contribution. Environmental effects on plant phenotype, hydrangea macrophylla, action of genes responsible for floral color is influenced by soil acidity. This is true. Flower color ranges from pink to blue. This is because of the environment. Now you are getting confused whether really the genes are doing the genes. Yes, they are interacting with the environment. The the soil acidity, right? What kind of the pH of the soil is there? You will get the same genotype will behave differently. So that's the environment effect. This bigger effect, hydrangea macrophylla. I'm just giving you the example here. Otherwise, we'll get there. Genetic studies, backbone of crop breeding. Imagine resume genetics. build the hypothesis, test what you have assumed is correct or not. You can go for chi-square test and then conclude, yes, it's imagination. You are not going to see any genes there, right? Now, of course, the molecular biology or genetic engineering now pointing better. Even the quantitative trial loci also addressing the polygenic. But here, the conventional way is, you want to study the genetics, you have to imagine okay, this is what you are seeing in the F2 generation. You have to assume whether it is controlled by one gene or two genes, maximum three genes. Beyond that, we need not. But even within the two genes, also so many combinations are there. Epistudies are there. Interaction between the non-allelic genes. So that you have to assume and build your hypothesis. Then whatever your hypothesis, whether it is correct or not, you have to conduct experiment and get the ratios and try fit to this chi-square test. And then if whatever that inference is there, whatever expected ratio is true or it is a chance factor, based on that you can conclude whether it is controlled by one or two genes or whatever that digenic ratio you have assumed. That is going with your hypothesis. Yeah, we were discussing about the terminologies. Um, different terminologies. What are the terminologies? Just want to have some look at, and then we can sort back to that distributing principles. Adaptation. What is adaptation? What is acclimatization? Allele or allelomorph, gene, locus, dominance, recessive, epistatis, genotype, phenotype, gene penetrance, expressivity of gene, ampeliploid, strenuploid, euploid, alloploid, autoploid, monoploid, haploid, diploid, so many terms. Then A synapsis and the synapsis, basic number chromosome, then the bivalent, univalent, certified seeds, foundation seeds, breeder seed, nucleus seed, truthfully labeled seeds, then centromere, kinetochore, chiasma, crossing over, linkage, coupling phase, repulsion phase, linkage, chromosome, chromatid, combining ability, diallele, dihybrid, single cross hybrid, double cross hybrid, three-way cross, triple cross, polycross, test cross, stop cross, reciprocal cross, drift, panmixia, all these duplex, nulliplex, simplex, triplex, quadruplex. What do you mean by that? The recessive polyploid throughout loci, and then the nullisomes, monosome, trisome, tetrasomes, prepotency, progeny test, protandry, protogyny, virulence, genia, metagenia, embryo and endosperm, maternal effects, pure line, inbred, transgressive. What now we use very many times that pure line and inbred where to use, what exact meaning of these terms, and transgressive segregates, rogue, off type, SIP, full SIP, or, and then half SIP. Okay, these are the terminologies what we want to discuss. I think yeah, just we discussed about the adaptation. What is the adaptation? The acclimatization. There's acclimatization. That's a process. Then adaptability. It's the capacity of the genotype to master the macro environmental factors to perform consistently. It's adaptability over time. It can be year, season, and the space, location, latitude. That's adaptive capacity of the genotype to master. So the terms are almost similar, but slight variation. Where you should use that term is important. Adaptation, acclimatization, adaptability. That's adaptation a process. Acclimatization is also a process, but it is the end, right? It is getting adapted to that. Then adaptability. So light change. Yeah, when you use the term adaptability, you said it, how it matches stability. So how we are different of this adaptability and stability. So adaptability is adjusting, right? Capacity is genotype to master the macro environmental factors to perform consistently over the time. So it is adapted to a particular location, season, you can say. Whereas stability means you have assessed the performance of the genotypes over the seasons or the locations, and you have brought out certain, the statistical measures to explain this is stable, this is not stable, this is highly, the sensitive, like that you bring up. So a slight difference from this adaptability and stability. Yeah. So that's what I just want to. Idiotype. Yes. What do you mean by this? Physiologically efficient model plant. That means your imagination. Physiologically efficient model plant type. A biological model of the plant, you can say, plant frame that can exploit the available resources most efficiently to produce maximum biomass, hence maximum economic yield. That is what idiotype. Always, but most of the time, how this plant should be. Many a times it's a model. To achieve that model practically, the breeder has to put his efforts. But just to decide what he wants, so that is the idiotype. What kind of idiotype means you have to define what should be the height, how many branches, how many leaves, how the structure of the plant, what other plant parameters also involved. You has to define, so that whatever you are defining, that plant should have this capacity. It's a physiologically efficient model plant type, a biological model of the plant frame that can exploit the available resources most efficiently to produce maximum biomass, hence maximum economic yield. That is what the idiotype. Allele or allelomorph, we said it, one of a pair of or a series of forms of a gene which alternative in inheritance because they are situated at the same locus in a homologous chromosome. So that is what allele or allelomorph. Same thing. Then gene. What do you mean by the gene? The unit of inheritance. So unit of inheritance, genes are located at fixed loci in a chromosome and can exist in series of alternate forms called alleles. You know how you can. So your definition, you should be very clear, not loosely talking about it. Your definitions should be very precise, complete sentence, meaningful sentence you have to use to define this terminology. Locus, the position occupied by a gene in a chromosome. Place, position. Dominance, it is intraallelic interaction. Remember, it's only between the within a gene. Intraallelic means between the alleles of the same gene. Intraallelic interaction, dominance, such that one allele manifests itself more or less when heterozygous, that its alternative allele, than its alternative. That means manifest means it expresses itself more or less, when heterozygous, than its alternative allele. So this masks the expression of the alternative. Recessive, the member of an allelic pair which is not expressed when the other member occupies on the homologous chromosome. Right? On the chromosome, we have two alleles, right? The one allele which is not expressing because of the presence of the other member, right? That is dominant member. It's not expressing on a homologous chromosome. This is very important. Homologous chromosome is very important here, right? Yeah, you have got a codominance. It is codominance with non-identical alleles. Specify two phenotypes that are both expressed in heterozygotes. So codominance means alleles are not same, they are different. They specify two phenotypes that are both expressed in heterozygotes. That means both these alleles are expressing. Because of that, you have got a different kind of phenotype. Right? Phenotype is going to be different one. It is not like your homozygous dominant genotype. Okay. So dominance means it's other one. So when you say codominance, yes, non-identical alleles specify two phenotypes that are both expressed in heterologous conditions. Genome, a set of chromosomes corresponding to the haploid setups. That is genome. And all the different types of chromosomes that particular species possess is there. That is called a genome. A set of chromosome corresponding to the haploid setup a species. Haploid setup. That's one. Then epistatus. Just like the dominance and the codominance is intrallelic interaction. Okay, intrallelic interaction. This is inter what do you call say between the genes, right? Between the genes. So epistatus means dominance of one gene over non-allelic gene. So one gene is interfering in the expression of the other gene. So dominance of one gene over non-allelic gene. The gene suppressed is said to be hypostatic, which is suppressing is hypostatic. More generally, the term epistatus is used to describe all types of interallelic, right, between the genes. Interallelic interaction, whereby manifestation at any locus is affected by genetic phase at any or all other loci, right? That's what the quantitative genetics. Yeah, it's being used, right? This epistatus used. Otherwise, these epistatus of digenic ratios, that is different. But whereas in polygenic, yes, many number of genes you are taking together and explaining. Genotype. What do you mean by the genotype? The entire genetic constitution of an organism is genotype. Genetic makeup of an organism is a genotype. Phenotype. Appearance of an individual as contrasted with its genetic makeup or genotype. So it's appearing. It is in proportion to that its genetic constitution. So also used to designate a group of individuals with a similar appearance but not necessarily identical genotypes. They look same, right? They look same but need not be genetically same. Okay, so phenotype. Penetrance. It refers to the gene penetrance, the frequency with which a gene produces a recognizable effect in individuals which carry it. Sometimes, the gene is there, it is not expressing. Sometimes it is expressing. That's what the frequency with which the gene produces a recognizable effect in individuals which carry it. So that is the gene penetrance. Then expressivity, the degree of manifestation of a genetic character. So it is the extent, that is the frequency. It is the intensity, that is the frequency. Remember, right? Maybe there are several individuals are there, few they will express and few they do not express. That is called a penetrance. Whereas expressivity, gene is there and the extent of that character of that gene is expressing. That's degree of manifestation of the genetic character, right? That is expressivity. You know the difference. It speaks about the intensity, that speaks about the frequency. Autogamy term is used to self-fertilization. The genome, set of chromosomes corresponding to the haploid set of species, already explained. Allopolyploidy, right? Haploid meningitis, again recollecting that genome. Allopolyploidy, a polyploid containing a genetically different sets of chromosomes. For example, sets from two or more different species, allopolyploid. Right? A polyploid containing genetically different sets of chromosomes. It is that now you are referring it to the species, other species, two or more different species that sets of chromosomes are derived. Allopolyploid. Autopolyploid, a polyploid arising through the multiplication, the complete haploid set of a species. So that means It is whatever it is there, it is copy. Haploid set, whatever it is there, it is the same thing is there. It's a copy, diploid. It's just haploid, same is it's same set is there. Same set of chromosomes are there. Now referring to the chromosomes, same set of chromosomes are there. Whereas allopolyploidy, one set from one species, another set from another species, like that. They are different, right? So within a species. So polyploid arising through multiplication of complete haploid set of species. Ampidiploid, a polyploidy whose chromosome complement is made up of the entire somatic chromosome of two species, ampidiploid. So it's a polyploid, right? Whose chromosome complement is made up of the entire somatic complement of two species. That means it is the diploid, right? But there are two species. One species, two copies are there. Another species also two copies are there. It becomes ampidiploid, right? So because if you hybrid, it's a good example you get in okra, where if you cross two different species, you will get, but its sustainability, further reproduction is, you will get that. The two different species you cross, you get the zygote. But to make it viable, to make it, the fertile, you have to double the chromosomes. So regular reproduction, meiosis can happen. The pairing chromosomes should be there. For that, you have to make two copies. So that is, there's each two species, whatever contributed in F1, that is doubled, and that becomes a ampidiploid, right? Because each species in diploid form. So that becomes ampidiploid. And it survives because of the regular, the cell division or meiotics or cell divisions. Entire, so mitotic also. Entire somatic complements of the two species is there. You do it for, for further reproduction. Aneuploids. So euploid and aneuploid. First, if you classify, of course, once you know that ploidy breeding, an organism whose somatic chromosome number is not an even multiple, because once you study, you know all that. But you have to define it. That's why terminologies I'm saying. Terminology is how you have to define. You should always independently, rather than while explaining you applied and you applied it, you will recollect. But straight away somebody asks you, what is aneuploid? Then you have to. An organism whose somatic chromosome number is not an even multiple of the haploid number. It is deviating from the even, right? Okay. So it is different. Euploids means all that, all of that MP diploid or autoploid, right? All that you're saying. It's exact set, complete set is multiplied. Chromosome is there in multiple forms. I mean, multiple copies rather. So aneuploid means some particular chromosome is not having the same number of copies of other chromosomes. So an organism whose somatic chromosome number is not an even multiple of the haploid number. So that's what your aneuploids. There are euploids, an organism whose somatic chromosome number is even multiple of the haploid. That's just opposite. Same set. I mean, it's copies, same number of copies. All chromosomes have same number of copies. That is euploid. Whereas aneuploid, some chromosomes are having two, some are having one, like that variation. Some are having three, some are having two, like that. That is aneuploid. What is existing in nature is different, but the definition here. Monoploid, an organism with the basic chromosome number, that is monoploid. Haploid is different. If haploid means at the time of meiotic, 50% of the chromosomes go to the different poles, you said, right? So there, that becomes a haploid. But that need not be, for example, tetraploid, the tetraploid is there. Some 48 chromosomes are there, totally haploid, I mean, tetraploid, like your potato. There, the haploid number is 24 becomes at the time of the cell division. 24 becomes. Whereas if you further divide again, that means there are two copies of each chromosome. That 24 means originally 12 only are there. So that is original is referred as a basic chromosome number. That means there is no further similarity within that set of basic chromosomes. Basic chromosomes, all the chromosomes are different from one another. Whereas in haploid, need not, right? It depends. If diploid is there, diploid basic chromosome number, if you are getting a haploid, it's same. From a diploid, if you derive a haploid, that will be same as a monoploid. Whereas monoploid meaning you derive it from any of the species, I mean any of the ploidies, but whose basic chromosome number it should have. Then only you can call it as a monoploid. You got a point. Haploid, if you get a, for diploids, if you are getting a haploid, it will be same monoploid. But from tetraploid, if you get a haploid, that becomes, two diploids, right? The gamete is forming with a diploid actually, but you call it as a haploid because 50% of the chromosomes are separated, forming into the gamete. So monoploid is different from that haploid. A cell or organism with a gametic chromosome number, that's all. In a gamete, what all the sets of chromosomes are coming, you are referring it as a haploid. Whereas monoploid, that organism having basic chromosome number. So there is no further similarity within that group as far as chromosome is concerned. Whereas in haploid, there is a possibility. There is a possibility. Some chromosomes can be, having two copies in that haploid. I explained it in tetraploids. It happens. Right? So diploid, an organism with two chromosomes of each kind. Each chromosome is having two copies. That is called as a diploid. Okay, so that is diploid. Heterokaryosis. The presence of two or more genetically different nuclei within a single cell of a mycelium fungi, normally you get it, is referred as heterokaryosis. Two different nuclei are there in the same cell, heterokaryosis. Somatic cell division, the mitosis, they have phases like prophase, metaphase, anaphase, and telophase. So that entire process is called cytokinesis, cell division. And then the mitosis, the nucleus division, or karyokinesis. Cytokinesis means cytoplasma division. Okay. Nuclear division or the karyokinesis. Cytokinesis means cytoplasma division. The meiosis or reduction division, right? The gamete formations. Mitosis is just a copy. Same cell is getting copied. Whereas meiosis, there is a reduction in the, because the gamete, are forming in the sexual reproduction. That is because of the meiosis only. Reduction division, that's a gamete formation. There, division 1, you have reduction division, extended prophase 1, and division 2, it's mitotic in nature. Division 2. Division 1 is one specialized, whereas division 2 is the same as a mitotic division. So that's why you call prophase 1, right? That is the division 1, whereas division 2, you can call that as a prophase 2, something like that. Prophase 1 is the extended one. You have got a leptotin, zygotin, packaging, diplotin, and dikinesis. So here it is extended form, and most importantly, zygotin, a stage in a meiotic prophase when the thread-like chromosomes appear. Okay. Packaging, the double thread stages of meiosis. No, you're saying that's getting into paired, same chromosomes, they come together. Okay. So then diplotin, the stage of meiosis which follows the packaging and during which 4 chromatids of each bivalent move apart into 2 pairs but remain attached at the region of chiasm. That means